Chapter 8 discusses nucleotides and nucleic acids. Here are the learning goals for this chapter. We're going to look at the biological function of nucleotides and nucleic acids, their structures, the structure of DNA and RNA, how DNA can be denatured, and the chemistry of nucleic acids. Nucleic acids are polymers of nucleotides, and they are used for a variety of things, including the storage of genetic information, such as in DNA, the transmission of that information, the processing of genetic information, and protein synthesis. And in their monomer form, they also have cellular functions such as providing energy for metabolism in the form of ATP. They can be enzyme cofactors, and they can be important in signal transduction. A nucleotide is composed of three things: a nitrogenous base, a pentose, and a phosphate. So over here you can see here in the box is the pentose, so that's a sugar. And then the purine or pyrimidine base, that's the nitrogenous base. It's called nitrogenous because it contains nitrogens, is attached at the 1' position. And then there's also a phosphate group shown here. A nucleoside is something that contains a nitrogenous base and a pentose, but not the phosphate. Carbon and nitrogen atoms on the base are numbered in a cyclic format shown here. And then the carbons of the pentose are designated with a prime to distinguish between the numbering on the pentose and the numbering on the base. So here this would be 1', that's the anomeric carbon. And here the sugar is in its cyclic form, and it's beta, so that's the beta anomer. And then this would be 2', 3', 4', and 5'. So here the phosphate group is attached at the 5' carbon of the pentose sugar, and the base is attached to the 1' carbon. The bases used are derivatives of pyrimidine or purine, which are shown here. These are nitrogen-containing heteroaromatic molecules, so they're aromatic, and they have nitrogen in them, not just carbon, which is how they get the term heteroaromatic. They are planar or almost planar, and they absorb light in the region around 250 to 270 nanometers. The two types of purines that we see are adenine and guanine, shown here. And then the pyrimidine bases are cytosine, thymine, and uracil. Thymine is only found in DNA, and uracil is only found in RNA. So the difference between these two is whether or not there's a methyl group at that carbon. There's not in the uracil, but there is in the thymine. And then cytosine differs in that it doesn't have this carbonyl. Cytosine, adenine, and guanine are both found in DNA and RNA, but thymine is only found in DNA and uracil is only found in RNA. All are good hydrogen bond donors and acceptors, and they are all neutral at pH 7. Now, in addition to those bases, you also have minor purine and pyrimidine bases. This slide shows some of the minor bases of DNA. 5-methylcytidine occurs in the DNA of animals and higher plants. N6-methyladenosine is found in bacterial DNA, and 5-hydroxymethylcytidine is found in the DNA of animals and of bacteria infected with certain bacteriophages. This slide shows some of the minor bases found in tRNAs. Notice here that pseudouridine, the only difference is the point of attachment of the ribose. It's at carbon 5, not the nitrogen. And then thiouridine has a sulfur instead of an oxygen. This table gives the different names for the base versus the nucleoside versus the nucleotide. So the bases are divided into either purines or pyrimidines. The purines are adenine and guanine. Now the nucleoside, which is the ribose connected to the base but without the phosphate, the adenine becomes adenosine, and then if you have the deoxy version, it's deoxyadenosine. And then the guanine is guanosine or deoxyguanosine. So the deoxy nucleosides are found in DNA, not RNA. With the pyrimidines, cytidine is the nucleoside, which has the cytosine pyrimidine base. And then when it's part of a nucleotide, the ending changes to YLATE. The pentose form differs in some nucleic acids and nucleotides. So the beta-D-ribofuranose is found in RNA, whereas the deoxyribofuranose is found in DNA. So this would be the open chain aldehyde form of ribose, and then when it forms a ring, it becomes furanose, and this is the beta anomer because this group is on the same side as the CH2OH group. And the numbering for this would be 1, 2, 3, 4, 5. That pentose ring can actually adopt what are called puckered conformations, where this carbon can either be a little bit above or a little bit below the plane of the sugar molecule. Here are the structures, names, and symbols for the deoxyribonucleotides. So notice it's deoxy because there's no OH group here. So it's lacking an OH group. So this first one is deoxyadenylate or deoxyadenosine 5' monophosphate. So we have one phosphate group attached at the 5' carbon of the sugar, and then we have an adenosine attached at the 1' position. And the symbols for this include A, Da, and DAMP. And the nucleoside, which would be this structure without the phosphate group, is just called deoxyadenosine. And then similarly, here we have deoxyguanylate, which contains a deoxyguanosine attached to a phosphate group. The symbols for this are G, DG, and DGMP. And the nucleoside, so the structure without the phosphate, is deoxyguanosine. Then here we have deoxythymidylate, also known as deoxythymidine 5' monophosphate. And here are the 1, 2, and 4 letter codes. And then here, deoxycetylate, also known as deoxycytidine 5' monophosphate. And here are the symbols for that. And here are the structures, names, and symbols for the ribonucleotides. So these are not the deoxy ones. They do have an OH group at carbon 2 of the sugar. Now the ribonucleotides actually have 1-letter abbreviations and 3-letter codes. Now you can also get other nucleotides where the phosphate group is attached to different carbons, and these are often formed by enzymatic and alkaline hydrolysis of RNA. In nucleotides, the pentose ring is attached to the nitrogenous base via an N-glycosidic bond, and the bond is formed to the anomeric carbon of the sugar in its beta configuration. The bond is formed to position N1 in pyrimidines and N9 in purines, and it's quite stable towards hydrolysis, especially in pyrimidines, but it can be cleaved if there's acid present. Now there's relatively free rotation around this N-glycosidic bond, and as a result, you can have a syn or an anti-conformation. The syn conformation has an angle near 0 degrees, whereas the anti-conformation, the angle is 180 degrees away from the sugar. These bases can actually tautomerize, and you have seen keto-enol tautomerism in ketones in organic chemistry. Well, we can also get tautomerism in these bases. So for uracil, it can exist in any one of these 3 tautomers, but which one predominates will depend upon the pH. And tautomers, as a reminder, are constitutional isomers that differ in their placement of hydrogens. So here, this hydrogen is now on the oxygen, and that pi bond has switched to be between this carbon and this nitrogen. And then here, this hydrogen is now on that oxygen in this tautomer version, and that pi bond has moved to be between that carbon and that nitrogen. Successive nucleotides of both DNA and RNA are covalently linked through phosphate group bridges called phosphodiester linkages, shown here. Thus, the covalent backbones of nucleic acids consist of alternating phosphate and pentose residues. And those phosphate groups are negatively charged at pH 7. In ribonucleotides, hydrolysis can occur under alkaline conditions where the 2' hydroxyl acts as a nucleophile in an intramolecular displacement. So a base can deprotonate the hydrogen on the 2' hydroxyl, then that negatively charged oxygen would attack the phosphate group and break that phosphodiester linkage. DNA, which doesn't have this hydroxyl group, would be stable under similar conditions. So we wouldn't see this type of hydrolysis with DNA. The functional groups of pyrimidines and purines are ring nitrogens, carbonyl groups, and amino groups. Hydrogen bonds involving the amino and carbonyl groups are the most important mode of interaction between two complementary strands of nucleic acids. So here are some examples showing the hydrogen bonding between two base pairs, between adenine and thymine, and between guanine and cytosine. And the hydrogen bonding is shown with the blue lines. And that's it for this video.